Good morning, class. Today's topic is DNA structure. Actually, the structure of DNA is known as double helix. Means there are two helical structure anti-parallel to each other. Watson and Crick was proposed the structure of DNA model in 1953. For this work, they got the Nobel Prize in 1962. According to Watson and Crick model of DNA, the definition of DNA is that DNA contains two polynucleotide strands coiled together in a helical manner. Hence, the name Watson and Crick double helix structure of DNA is given by them. Here is a photograph of James Davy Watson and Francis Harry Krypton Crick. According to their model, the structure of DNA is a double-stranded polynucleotide molecule having sugar and phosphate forms the backbone, and the bases or nitrogenous bases projected to inside the area. The two strands are coiled each other and arranged in anti-parallel. Means if one strand has fifth to third end, then the other have third to fifth end in the direction. The common diameter between the two strands of DNA is 20 angstrom. Adenine of one strand pair with thymine of another strand, and it joined with the help of two hydrogen bond and vice versa, and guanine of one strand pair with cytosine of another strand by 3 hydrogen bond. Because of a complementary base pairing, if one strand of a polynucleotide sequence is known, another can be detected. If you know about the one strand of polynucleotide sequence, then you can detect the another strand base pair. For example, if we are going from fifth end to third end, look at the sequence A, G, T, 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 A, C, A, T, A, C, C, G, G, A, 4 times, then T, T, and so on. Then the another strand have the opposite base pair. For example, in the position of adenine there is a thymine. In the position of guanine there is a cytosine. So the complementary strand, if twisted each other, then what is the distance between these two strands is 34 angstrom. And in each twist, because it's a twisted structure, so in each twist the DNA contain near about 10 base pair always, and the distance between these two base pair is 3.4 angstrom. Here is the anti-parallel strand of DNA structure. You can see in this picture the groups is there and twisting is there, nitrogenous base pair is also there, and sugar phosphate backbone is also there in the picture. This is the another picture. You can see the 34 nanometer in between the two twistings, 0.34 nanometer. The structure of phosphate bonds, hydrogen bond, and the major groups in between the two twistings. Now, what is the meaning of packing? Means it is a very long nucleotides molecules, polynucleotides molecules. So how it is found in a nucleus? What is the packing phenomena or packing type of this DNA in the eukaryotic cells or eukaryotic nucleus? Actually, DNA is stabilized with the positively charged, because the DNA is a negatively charged particle. So it have to be stabilized with the positively charged basic protein, and that protein is known as histone protein. Histone are positively charged. Why it is positively charged? It is due to rich in the basic amino acids like lysines and arginines. Histones are organized to form a unit of 8 molecules. There is the 8 molecules of histone protein organized in a form. So this structure is known as histone octamer. Negatively charged DNA wrapped, it is in a form of a wrapping around the positively charged histone octamer to form a structure. That structure is known as nucleosome. And under the electron microscope, if we are going to see this nucleosomes, we find that are seen as beads on a string structure like. Nucleosomes forms the repeating unit of a structure, that nucleus is called the chromatin. A thread-like structure known as the chromatin. Now what is the chromatin? Chromatin is packed to form the chromatin fiber. And these are again further coiled and condensed. At what phase? At metaphase stage, to form the chromosome. Now there are two types of chromatin threads. One is the euchromatin, another is the heterochromatin. Euchromatin in the nucleus, some loosely coiled region of chromatin thread. If we are going to stain that chromatin thread and see under electron microscope, we see that there is a light strand structure, and light strand structure is known as the euchromatin. And in the nucleus, more densely packed regions of chromatin, it is stained dark in color, and it is known as the heterochromatin. When we are going to study the transcription, we find that euchromatin is transcriptionally very active in comparison to the heterochromatin, because it is a loosely coiled region of chromatin thread. So this is all about the structure of DNA seen under electron microscope. In the next class, we will study about what is the transforming principle. It is the next topic of your class. So class, thank you and have a nice day.